السلام عليكم ورحمة الله وبركاته. أرحب بكم بالعام الدراسي الجديد وفي هذا الفصل الجديد. أتمنى أنه كلكم تكونون أنتم عوائلكم بخير وسلامة. Our topic for today is the histology of the skin. The skin is the largest organ in the body. It accounts for about 20% of total body weight, and it has a surface area of about 2 meters squares. The skin is also called the cutaneous layer, the cutaneous membrane. It is also called the integument. And the word integument means the outer covering of an organ. So the skin is the outer covering of the human being. It is the boundary between the internal and external environment of the body. Now the skin is formed of the epidermis and dermis, so it is formed of two layers. A fatty layer called the hypodermis, the subcutaneous layer, connects the skin to the underlying tissues. Now, in addition to the epidermis and dermis, we have other structures that are found in the skin, and these include sensory organs, the erector pili muscle, hair, nail, sebaceous and sweat glands, and the mammary glands. Now, actually, the hair, the nail, the sebaceous and sweat glands, and the mammary glands are epidermal derivatives. They are derived from the epidermis, as we are going to see in the subsequent slides. Now, this is an image of the skin. We can see here the skin is formed of an epidermis, the superficial layer, and a deeper layer, the dermis. And under them we have the hypodermis, the subcutaneous layer. In addition, we have other structures that we have talked about and that we are going to study each one individually. First, we start with the functions of the skin. The skin has several functions, usually grouped into several categories. Start with the protective function. The skin, first of all, is a physical barrier. It is a solid boundary that prevents or that protects the body against thermal and mechanical stresses and against several harmful substances. It protects the skin, protects the body from harmful microorganisms because there are certain immunological factors in the skin that we are going to talk about also. A very important function of the skin is protection against ultraviolet light. Ultraviolet light is both beneficial and harmful to the body. It is beneficial in a certain way and harmful in another way. We are going to talk in more details about that. But the skin provides a way to protect the body from the harmful effects of ultraviolet light. A very important function of the skin is that it prevents the excess loss of water through the skin. Why is that? Now, actually, the skin has a lipid-rich layer. There is a lipid-rich layer in the skin, and this will make the skin a selectively permeable membrane. Anything that is soluble in fat, lipophilic substances, can pass through this lipid-rich layer and therefore can pass through the skin. And this is why, and this is the basis why we can use certain drugs in the form of creams and ointments on the skin, and they can be absorbed because these substances are fat soluble. They can pass through this fat. However, water-soluble substances and water cannot pass it through this lipid-rich layer, and therefore the skin prevents excessive loss of water. Now, actually, this is very, very important because This allowed life to become terrestrial. Now, living on land, terrestrial life, depends on independence from water. So an organism must preserve its own water inside its body, and this can be done by the skin. Without it, the organism must stay in the water, must spend a lot of time submerged in water. Now, if someone has extensive burns and loses his skin, he will lose a lot of fluid through the skin because this protective function of the skin has been lost. This lipid-rich layer has been lost, so a lot of fluid will be lost. So one of the first steps in the treatment of burns is to replace this fluid that has been lost. The skin also has sensory function. Because the skin is in contact with the external environment, it has a large surface area. It is a good place to receive stimuli from the outside. And in the skin we have mechanical receptors, we have thermal receptors, we have receptors for pain, and therefore it plays a very important sensory function. Another function of the skin is the thermoregulatory function of the skin. It regulates body temperature. By insulation. The subcutaneous layer, which is mostly fat, provides insulation that preserves the heat of the body. Sweating plays an important role in thermoregulation because sweat is mostly water. When it goes on the surface of the skin, it evaporates, cooling the body. Also, there is superficial microvasculature in the skin, arranged in a specific way that helps in thermoregulation. We are going to talk about that in the next slides. Metabolic functions. Several metabolic functions occur in the skin. One of the most important is the synthesis of vitamin D3 through the action of ultraviolet light. So we need ultraviolet light to synthesize vitamin D3. Also, excretion of excess electrolytes with the sweat. And finally, storage of energy as fat. And this happens in the subcutaneous layer. As we have said, it is formed of fatty tissue. So now we start with the first layer of the skin, the superficial layer, the epidermis. The epidermis is a stratified squamous keratinized epithelium formed mainly of epithelial cells. The epithelial cells of the epidermis are called keratinocytes, and these keratinocytes are actually arranged in layers. The keratinocytes, they are arranged in layers, and very importantly, the keratinocytes are connected with each other, adhere to each other by desmosomes. This is very important. In addition to keratinocytes, in the epidermis, we also have non-epithelial cells, melanocytes, Merkel cells, and Langerhans cells. These are non-epithelial cells, but they perform other functions in the epidermis. Now, according to the thickness of the epidermis, we classify skin into thin and thick skin. So when we say thin skin or thick skin, we are talking about the thickness of the epidermis. Now, in thick skin, for example, the skin that is found in the palms and the soles, the epidermis is thicker and it's formed of 5 strata. It is formed of 5 layers. Elsewhere in the body, the epidermis is thinner and it is formed of only 4 strata, and it is called thin skin. So thick skin is formed of 5 strata. The epidermis is formed of 5 strata, whereas in thin skin, the epidermis is formed of 4 strata. Now, total skin thickness means the combined thickness of the epidermis and dermis, both of them. فإذا نقول thick أو thin skin فإحنا نتكلم فقط عن thickness of the epidermis. بس بنتكلم عن total skin thickness فإحنا نتكلم عن the epidermis and the dermis. Of course, this varies according to the region of the body. Total skin thickness. Total skin thickness is thickest on the back and it is thinnest on the eyelids. So it is thickest on the back, thinnest on the eyelids. So here we have, this is the epidermis. All this is the epidermis. Here we have several layers. This is a layer, it's another layer, here's another layer, and these are all the layers of the epidermis. These are all keratinocytes. These are all keratinocytes. In addition, here we have melanocyte, Merkel cell, and Langerhans cell. And we are going to talk about the characteristic of each of these layers, the characteristic of each of these cells. So now let's talk about the layers of the epidermis. And we start with the stratum basale. Stratum basale is the deepest layer of the epidermis. It lies directly on the basal lamina. This is, here in this image, this is the stratum basale. Now the stratum basale is formed of only a single layer of cells, and these cells are basophilic. These cells are basophilic, and usually they are cuboidal. They are cuboidal. Now, the cells of the stratum basale are adhered or are connected to the basal lamina by hemidesmosomes, and at the same time, they are connected to other cells of the stratum basale and to the cells of the adjacent stratum spinosum by desmosomes. This is very important to keep the cells of the epidermis connected with each other so that they will not separate. The cells of the stratum basale are highly mitotic cells. They are highly mitotic cells, and they are considered the progenitor cells of all the other layers. So the cells in the stratum basale will form all the other layers of the epidermis. As you can see here, for example, here is a dividing cell. This is a cell from the stratum basale that is dividing. The resultant cell, one of them will remain inside. So here is a dividing cell. One of the cells will remain in the stratum basale. The other daughter cell will pass upwards or superficially into the stratum spinosum. And then, as this cell becomes more mature, it will continue to migrate upwards or superficially until it reaches the other layers, until finally it reaches the superficial part of the epidermis, and here it will be shed. This process from the birth of a cell until it is shed, the turnover of the cell takes about 15 to 30 days, meaning it takes about 2 to 4 weeks, meaning that the epidermis is renewed during this time. A characteristic feature of all keratinocytes is the presence of a special type of intermediate filament called keratin. And this is why they are called keratinocytes. Then we'll have the stratum spinosum. The stratum spinosum is usually the thickest layer of the epidermis and is even thicker in the thick skin. So this is the stratum spinosum. When we said in thick skin we have a thick epidermis, one of the reasons for the high thickness of the epidermis in thick skin is the high thickness of the stratum spinosum. Now, the stratum spinosum is formed of several layers of polyhedral cells that are active in keratin synthesis. So these cells are active in protein synthesis and they have all the features of protein synthesizing cells. Now, the cells of the stratum spinosum that are close to the stratum basale are also mitotically active. So this is the stratum basale in the image here. These cells that are adjacent to them, these are also mitotically active. Together, these two layers together are called the stratum germinativum. So the deepest part of the stratum spinosum with the stratum basale are both of them mitotically active. Both of them help in forming all the layers of the epidermis, and together they are called the stratum germinativum. Now, the keratin filaments in the cells of the stratum spinosum become thick tonofibrils. They become thick tonofibrils. And these tonofibrils are attached to desmosomes. So if you look at the electron microscope image here, these are the desmosomes. These are desmosomes. And these thin black lines, these are the tonofibrils, the keratin tonofibrils. Notice how they are attached to the desmosomes, of course, from each side. Now, when we prepare a tissue for a histological study, the cell will shrink because, as you know, the cytoplasm is mostly formed of water. It will evaporate and the cytoplasm will shrink. But remember that desmosomes and the keratin tonofibrils are protein structures and they do not shrink and they remain intact. Now, this will make the desmosomes and the tonofibrils appear as spines on the surface of the shrunken cells when we examine them under the microscope, and therefore they are given the name stratum spinosum. If again we look at the electron microscope image, this is the cell. This is the cell. And notice here how here we have the spines. These are the spines. Why? Because it is formed from the desmosomes and the tonofibrils that do not shrink, whereas the cytoplasm here has shrunk. In the light microscope image, we can see that this is a cell, and we can see that on the surface of the cell I have small projections, and these are the spines, and therefore it's called stratum spinosum. Stratum granulosum. Stratum granulosum, this layer here, is formed of few layers, maybe 3 to 5 layers of cells, and from the name granulosum, these cells have granules, and they have two types of granules: keratohyalin granules and lamellar granules. The keratohyalin granules contain keratin tonofilaments associated with proteins. So here we have the addition of some new proteins to the structure of the tonofilaments. These granules are non-membranous. They are not surrounded by membrane. Why is that? Because actually the content of these granules, the keratin tonofilaments that are associated with proteins, they are used inside the cells. They remain inside the cells and they're used inside the cells, and therefore they do not require membranes. The keratohyalin granules appear as basophilic structures under the light microscope. So when we look at the microscope, we see cells with several dark dots inside them, And this is why it is called stratum granulosum, because the cells appear granulated under the light microscope. Lamellar granules, on the other hand, contain various lipids. They contain various lipids. And these granules are membranous. They are surrounded by membrane. Why are they surrounded by membrane? Because the contents are released to the outside of the cell by exocytosis. So first of all, we must remember two things. These are lipids. Lipids cannot be free inside the cytoplasm because, as you know, cytoplasm is mostly water, and lipids cannot be with water. So the lipids must be surrounded by a membrane, and then this lipid is released to the outside by exocytosis. Now, actually, these granules, the lamellar granules, are small, and they are seen usually by the electron microscope. In this image here, small image, they appear as oval structures with several lamellae. This is why they are called lamellar granules. So here, these are the lamellar granules in this image here. Notice how they are oval and they contain several lamellae. The lipids of the lamellar granules are released to the outside by exocytosis, as we can see here. So the lipids will be released to the outside and it will fill the intercellular space with lipid. This will create the lipid-rich hydrophobic barrier that prevents water loss that we have talked about in the beginning of the lecture. So this is the layer where the protective lipid-rich barrier is formed. It is the stratum granulosum. Before it, the layers before it, we do not have the lipid protective layer. Stratum lucidum is a translucent layer found only in thick skin. This is the stratum lucidum in the image here. Usually under the microscope they appear as colorless region, a very thin colorless region, and they are found only in thick skin, They are formed of eosinophilic squamous cells with no nuclei or organelles. So now the cells have no nuclei and no organelles. They are dead cells, and the cytoplasm is filled with keratin. This is why it's eosinophilic, because it's filled with the protein keratin. The last layer is the stratum corneum. This is the most superficial layer. It is a thick layer and is thickest in thickest skin. It is usually formed of about 15 to 20 layers, and these layers we have thin squamous cells which are given a special name, squames. The cells in the stratum corneum are called squames. These cells have no nuclei, no organelles. They are just cell membranes filled with keratin. So these are dead cells. These are dead cells. The cells in the deepest part of the stratum corneum are also surrounded by the lipid-rich barrier produced by the stratum granulosum. So the lipid-rich barrier is formed in the stratum granulosum, but it surrounds the cells up to the deep layers of the stratum corneum. On the other hand, the cells in the superficial layers of the stratum corneum will be shed to the outside. So, and these cells are replaced by cells from the deeper layers. Calyces are areas of thickened stratum corneum due to repeated friction. So areas of the body where we have repeated friction, the stratum corneum will become thickened to form calyces. And here's an example in this image. As most of us use pens and pencils, most of us will have such calluses in our middle finger, because this is the place where the pen rests and moves, producing friction, and this will lead to the formation of callus. Now, we have talked about the layers of the epidermis, and all these layers are formed of epithelial cells, keratinocytes. There are other cells in the epidermis, non-epithelial cells, non-keratinocytes. And the first one I'm going to talk about are melanocytes. These are neural crest derived cells, so these are derived from the neural crest. They are found in the stratum basale. Okay, so in the epidermis, the melanocytes are found in the stratum basale. Of course, they are also found in the hair follicle. We are going to see that in a few slides. And melanocytes produce the pigment melanin, which could be black in most people, and sometimes in certain individuals it could be red. You know, the people with red hair, these people actually produce red melanin pigment. Now, the cell body of melanocyte is pale staining. It is pale staining. It is found between the keratinocytes of the stratum basale. It is not attached to them by desmosomes. Remember when we said that the cells of the stratum basale are attached by desmosomes to other keratinocytes in the stratum basale and spinosum, but the cell body of the melanocytes are not attached by desmosomes to the other cells. However, melanocytes are attached by hemitosmosomes to the basal lamina. Now, the cytoplasm contains mitochondria, rough endoplasmic reticulum, and Golgi apparatus. In general, these are all features of protein-producing cells because melanocytes produce protein. Now, several processes from the cell body extend between the cells of the stratum basale and spinosum. Now, this is important. Melanin inside melanocyte is stored inside granules. The pigment melanin is produced in the cell body, and it is stored inside granules called melanosomes. So the melanosomes are formed inside the cell body of melanocytes. Then the melanosomes are transported to the processes of melanocytes. As we have said, these processes are found between keratinocytes, and the tips of these processes will be engulfed. They will be engulfed by the keratinocytes. They will be engulfed by the keratinocytes. The melanosomes will then form a supranuclear cap. They will spread to form a layer just above the nucleus. Now, this layer is formed of the dark pigment melanin. This will help to protect the DNA of keratinocytes from the harmful effects of ultraviolet light, because this pigment melanin does not allow ultraviolet light to pass. So this is the function of melanosomes. So if you look at this image, here, this is the cell body of melanocyte found between the keratinocytes of the stratum basale. The processes, however, extend between the keratinocytes of different layers. Now, melanosomes are produced in the cell body of melanocytes, but they are then transported through the processes and then taken up by keratinocytes. Now, although produced by melanocytes, melanin is found more in keratinocytes than in melanocyte. Okay? So it is produced by melanocytes, but it is mostly found in keratinocytes, melanin pigment. Now, a melanocyte and its associated keratinocytes are called the epidermal melanin unit. So, for example, this melanocyte with all the keratinocytes, it gives melanin 2. These represent an epidermal melanin unit. The density of this unit is the same in all individuals, whether it is Caucasian, white Caucasian person or an African person, it is the same density. Now, what differs between them? They have a Caucasian person and African person have different skin color. It is the rate and amount of production that differs. Even a white person, when exposed to sun for a long period of time, for example, when a person goes out to the beach, the density of this unit will not increase, but the production of melanin and the amount of melanin produced is going to increase when we are exposed to the sun. So as we have said, melanin plays an important role in determining the skin color, the color of the skin, but it is not only melanin pigment. Skin color is actually the result of several factors. The most important is melanin pigment, which is usually black. Keratin pigment, another important factor. Keratin pigment is usually an orange pigment. Also blood in the vessels. Blood in the vessels. Remember, there are no blood vessels in the epidermis, but the blood vessels are found in the dermis. So what we are talking about are the blood in the dermal vessels. So how much blood is found in these vessels? This is also determines the color of the skin. What is the state of oxygenation of the blood in these vessels? As you know, the color of oxygenated blood differs from the color of deoxygenated blood. So all these factors play a role in determining the color of the skin. Other non-epithelial cells found in the epidermis include Langerhans cells. These are antigen-presenting cells that are mainly found in the stratum spinosum, for example, in this image here. They have processes that extend between the different layers. These cells are antigen-presenting cells. So any foreign particle, any antigen that enters the skin, it will be picked up by Langerhans cells and presented to lymphocytes, and therefore they play an important role in the immune system, the immune response to foreign particles. Merkel cells, other cells are Merkel cells. These are mechanoreceptors for light touch, and they are mostly found in the fingertips. Now, Merkel cells are found between the cells of the stratum basale. So here we have the stratum basale, and here we have a Merkel cell found between them. Now, Merkel cells have cell body and with processes. Now, the cell body and the processes are attached to adjacent cells by desmosomes. So, attached to keratinocytes by desmosomes. The cytoplasm of Merkel cells contain neurosecretory granules. So they have neurotransmitters, and adjacent to them, adjacent to the basal surface, we have the expanded terminal of an unmyelinated nerve fiber. So here we have a fiber. Near its termination, it is expanded, and it lies close to Merkel cells. Now let's talk about the dermal-epidermal junction. The place where the epidermis comes in contact with the dermis, this is called the dermal-epidermal junction. Now a basement membrane is found between the epidermis and the dermis, of course, and the epidermis is attached to this basement membrane by hemidesmosomes, And the dermis, on the other hand, is attached to this basement membrane by collagen anchoring fibrils. So each part is attached to the basement membrane, and this will make the junction stronger. Now, actually, the surface of the dermis in contact with the epidermis is not straight. It's irregular, as you can see here in this image. Notice how this is the dermis. It's not regular, but we have several projections. We have several dermal projections called dermal papillae. These dermal papillae interdigitate with downward projections from the epidermis. These downward projections from the epidermis are called the epidermal ridges. So we have dermal papillae going up, and we have epidermal ridges going down, and they interlock with each other, they interdigitate with each other. Now, this interdigitation is very prominent in thick skin, and sometimes it's very prominent that it is shown on the surface of the skin as a pattern of grooves and ridges, and these are actually what we call the fingerprints, Now, why is this important? So why is this jigsaw-like arrangement of the junction between the epidermis and dermis important? It is important because first it increases the surface area of contact between the epidermis and dermis, thus reinforcing the junction, making the junction between them stronger. Second, it increases the number of Merkel cells and Meissner corpuscles, thus increasing tactile sensitivity. Now, as we have said, Merkel cells are found within the stratum basale. Meissner's corpuscles are found in the dermal papillae. Because of this arrangement, there is increase in the surface area. This means that we have increase in the number of Merkel cells and Meissner's corpuscles, making that region more sensitive to tactile sensations. Now, as we have said sometimes, this interdigitation is reflected on the surface of the skin. This will lead to increase in the surface area of the grip of the hand or feet by increasing friction. Because we have a series of grooves and ridges on the surface of the skin, the surface area that is used for gripping objects by the hand, for example, is increased. This means that there is more friction between the skin and the object. This means that we'll have a firmer grip. Although not exactly a function, but also a benefit from this arrangement. Now, the ducts of sweaty glands open on the tops of the surface epidermal ridges at what we call sweat pores. So in these ridges that are found on the surface of the skin, at the top of these ridges, the sweat glands open at pores called the sweat pores. Now the sweat and the ridges will form the fingerprints. Of course, or the footprints, when a smooth object is touched. So when we press our fingers on an object, because the sweat is on the surface and because we have a pattern, this pattern will be printed on the surface that we have touched. Since the pattern of the ridges is genetically determined, this can be used to identify an individual. So fingerprints are unique because they are genetically determined. So only in twins, for example, they are identical, but in different individuals, the fingerprints are different. And this can be used to identify a person. So all of these are benefits that come from this arrangement between the dermis and epidermis. So now let's talk about the dermis. The dermis is a connective tissue layer found under the epidermis. It is variable in thickness. It is thickest on the back. Remember when we said that The total skin thickness is most on the back, and this is because the dermis is very thick on the back. It is actually formed of two layers with no distinct boundary between them. The first is the papillary region. It is a thin superficial part of the dermis. It corresponds to the dermal papillae. So this is the dermis. This part that corresponds to the dermal papillae, this is what we call the papillary region. It is formed of loose areolar connective tissue containing blood capillary loops, lymphatic vessels, nerves, and importantly we have Meissner's corpuscles or corpuscles of touch found in this region. The reticular region of the dermis is the thick, deep part of the dermis. It is formed of dense, collagenous, irregular connective tissue. Dense, collagenous, irregular connective tissue. It contains collagen and elastic fibers, contains lymphatic and blood vessel plexuses, contains sensory receptors and nerves, hair follicle, erector pili muscle, and glands. So many structures are found within this region of the dermis. We are going to talk about them, of course, in the next few slides. Now, we said that the reticular region of the dermis is formed of dense, collagenous, irregular connective tissue. However, in certain regions of the body, the collagen fibers within the reticular region tend to orient more in one direction than another. Although the collagen fibers pass in all directions, but many collagen fibers in that region tend to pass in one direction more than the others. Why? Because of the natural tension that is found in that region. In that specific region, the forces applied are in a certain direction. From where does this tension come? The bony projections, orientation of muscles, movement at joints, all these contribute to this tension. So for example, if you look at this image here, in this region, this magnified here, because of the natural tension lines from bone projection or muscles, most of the forces are in this direction. Most of the forces run in this direction. And because of that, collagen fibers run in this direction. Now, this form what we call tension lines. Now, surgical incisions made parallel to these tension lines are associated with faster healing and less scarring. Why is that? So let's look at this part here. As we have said, these are the areas of tension. So the forces run in this direction. The forces run in this direction. The forces in this region pass in this direction. If we made an incision perpendicular to the tension line, like shown here in this image, because of the force are passing in this direction, the edges of the incision will be pulled in the direction of the force, meaning that they will be pulled apart. This will make the healing process take longer time, and it may result in scarring. If we look at this image here, these are the direction of the tension lines, meaning that this is the direction of the force. If I make an incision parallel to the tension line, this means that the incision will be pulled in these directions. This actually means that the edges of the incision will be closer with each other, will be tighter. This means that the healing process will be faster and less scarring will be formed. So now let's talk about the vascular plexuses found in the dermis. In the dermis, we have two major plexuses. The subpapillary plexus It is located between the papillary and reticular regions. Here, between the papillary and the reticular regions of the dermis. From this plexus, several papillary loops, arise into the dermal papillae, and these are important to supply nutrients for the epidermis. Then we have the deep dermal plexus, here, found in the deeper regions of the dermis. Now, between these two plexuses, we have anastomoses. These anastomoses, as you can see here, all these anastomoses are important for the thermoregulatory function of the skin. In case we have cold weather, it is cold conditions, the blood flow to the subpapillary plexus is reduced. This will reduce heat loss, and therefore the body will preserve its heat. On the other hand, if we have hot condition is hot, blood flow to the subpapillary plexus will increase, and this will lead to increased heat loss, and therefore the body will be cooled. On the other hand, the lymphatic vessels begin in the dermal papillae, and they will form plexuses that correspond to the blood vessel plexuses that we have talked about.